All right, so this video we're going to be going over the translation portion of protein synthesis. If you haven't watched the other videos, please go back and watch those. The translation is going to be about the part where you're actually building your protein. So just to remind you, we got transcription, translation. This video is about how your polypeptide is actually going to be built. So we can't talk about translation without at least mentioning ribosomes. So one big misconception is that a lot of students get confused and they think that the ribosomes just like arbitrarily kind of spit out a protein. It's not really the case. So let's kind of remind you guys about what these ribosomes do. And there's really going to be three sites that we're going to talk about. There's going to be what's called the E site, the P site, and the A site. So it's really going to be the E, the P, and then the A. I'll show you a better picture in just a minute, but a couple things about ribosomes is that they are made up of ribosomal rRNA. That's what that R in front stands for, and some proteins. They're made in the nucleolus, so that kind of center portion of the nucleus. So that's essentially where they're synthesized. It's going to be made actually in the nucleus and the nucleolus. So understand that. And basically ribosomes, they're going to basically be the site of protein synthesis. It's also abbreviated, yes. Okay. So yeah, they most definitely help aid in the process of protein synthesis. So ribosomes, they help make proteins, but by themselves, they don't really make proteins. There's going to be this whole kind of relationship between the mRNA and your amino acids and how they're brought over. So just understand, really, when we get into translation, you're building this, but there's a couple of key players involved. So if we're talking about translation, we also can't not mention these things in turquoise. These are going to be called your transfer RNAs, or abbreviated as tRNAs. Okay. Now, one thing that we need to mention about these tRNAs and what they do is there's a couple of things here. First and foremost, this thing in brown is the ribosome. So we have the ribosome, we have your mRNA in red, and it's going in this direction. The reason why I know it's going in this direction, meaning that it's reading in that direction, is that the 5' end is always fed in first, okay? Remember, the 3' end has that poly A tail, so you have a bunch of adenines over here. Now, these things in purple are each individual amino acids. Now, one thing that you're going to be in really good shape is, if you can catch this sooner than later, is that for every 3 nucleotides or 3 RNA molecules, codes for 1 amino acid. Every 3 nucleotides, which we're going to be calling a codon, is going to code for 1 amino acid. So this is showing you your growing protein or your growing polypeptide, okay? And we're going to go through this motion kind of everything that's going to be happening all in one step. So let's kind of take a closer look. So like I said, we're going to mention ribosomes. Ribosomes are going to help aid in the process of protein synthesis, as well as these tRNAs. So transfer RNAs, let's talk about what those are first. So what do these tRNAs do? And there's a couple of things that we need to at least fixate on. tRNA stands for transfer RNA. But basically the point of these is that they're going to be carrying amino acids to the ribosome. Okay. Now, tRNAs, one end contains what's called the anticodon, which pairs with the codon portion of the RNA. So I'll show you what that looks like in just a minute, but a codon is going to be the triplets that are going to be read by your tRNA, all right? Now, one big, massively important thing that I need to make known now is that each tRNA is specific to the amino acid it carries. So each tRNA is going to be specific to the amino acid it carries. Okay. Now, on one end, you're going to have over here, it's showing the anticodon. So that's going to bind with your codon region. And each tRNA is carrying an amino acid. So on the amino acid tRNA synthase is not something you have to know, but let's at least kind of look here. Amino acids, so let's say you had a meal with protein. Your body is going to basically metabolize it, break down those amino acids. And you just have to know nothing is just random. Nothing is by chance. So what I like to emphasize here is that how does this tRNA know which amino acid it's going to carry? Well, there's an enzyme called aminoacyl tRNA synthetase. And it just shows you that it gives the amino acid onto your quote unquote charged tRNA. When it has this amino acid, it's going to go and carry it to a ribosome to build a protein. Okay, so each tRNA is specific to the amino acid it carries. And so if you're still not able to put this story together in your head, let's kind of go back here. These things in purple, amino acids, like these cartoony things, realistically, to make it legit, this video, it's not like this purple kind of hexagon looking thing. It's carrying one of these. So that's what you need to understand. The amino acids we're talking about is one of these. So that's going to be your final protein. So jumping back here, each one of those little amino acids really, in all actuality, is looking like this. Okay, so just kind of keep that in the back of your mind as we kind of go through this visual. So let's kind of emphasize where we're at now. So hopefully you understand what tRNAs do. They're going to be carrying amino acids to the ribosome, and each tRNA is specific to that amino acid it's going to be carrying. Okay, so let's get into translation. As we mentioned earlier, transcription in the previous video was talking about, well, how did this mRNA sequence get made? You cannot, and I cannot stress this enough, you cannot do transcription, you cannot do translation without doing transcription first, meaning this mRNA sequence had to come from the DNA. The DNA had to be read, meaning the DNA had to be transcribed to make mRNA. Now this mRNA, the sequencing is going to be read to make this protein. So the end goal of all of this is making this purple thing, this polypeptide. This will become your protein. So we can kind of see here now, we have an empty tRNA. It's going to go grab another amino acid. Then these tRNAs are going to bring the correct one over. Okay. So what I mean by each tRNA is specific to the amino acid it carries, this is the anticodon. This CCG is carrying glycine or GLY. That's the amino acid it's carrying. AAA, this tRNA was carrying phenylalanine or PHE. That's the amino acid that one carries. So you're seeing now, hopefully, at least the relationship that the mRNA is being read and you're building your polypeptide according to what these sequences of codons are. So making a protein, very similar to transcription, in that there's 3 steps. We got initiation, elongation, and termination. So let me show you the visual real quick first, and then we'll kind of go back to the slides. And let's just jump to translation. So we've already made your mRNA. We processed it if it was eukaryotic. So, right, we cut it up. And now we're going to take that mRNA sequence. This is my 5' end. You see a bunch of tRNAs. I'm actually going to pause this. Notice I got a tRNA carrying an amino acid, tRNA with an amino acid. So this is my messenger RNA sequence, and let's just play it out and see what happens here. Notice I have a ribosome that's going to come. The base of my ribosome carrying a tRNA. Then the second portion of the ribosome is going to come, and basically now inside this ribosome we can see a couple of things. This is my tRNA. This is my first amino acid. We'll talk about all amino acids have to start with this methionine, MET. So this is AUG on my mRNA, carrying 1 amino acid. This is going to be my next building polypeptide. So let's see what happens here. I have another tRNA. Now watch what happens. These guys are going to form a bond right here, and then the whole ribosome is going to shift over. Now, notice this empty one leaves. A new one is going to come in. So I have a new charged amino acid being brought over. I'm going to form a bond here. And then the whole ribosome is going to shift over. This is going to be an empty tRNA. So you see this is your growing polypeptide. So this is taking a closer look at your mRNA. So these things in red, that's the RNA. So each triplet, GUC, that's going to be, actually, let me jump back. Each one of these is called a codon. That's a codon, and it bonds with the anticodon portion of this tRNA. So this would be your anticodon putting on the correct amino acid, and this is your growing polypeptide. So there's your amino acid, and you can see that this is the building of your protein. So GUC has the codon, bonds with the anticodon, and you can kind of see now there's a couple of sites about the enzyme that we got to at least address here. This would essentially be called your P site right here in the middle, because this is your growing peptidyl site, okay? That's your A site, and now this would be your E site. So let's kind of take a closer look now at what this looks like. So before I go through this, making a protein, let's kind of get this down first, okay? If you notice on that video, it showed that the mRNA bound to a start of a ribosome, not a full ribosome, but the smaller subunit of that ribosome. So if you take a closer look here, let's kind of at least address what's happening here. All coding regions. Now notice I had a bunch of RNA that didn't even get read. That's okay, that's gonna happen. Don't let that stress you out. So these first codons, or that first codon is always gonna be AUG, okay? So a simple way to remember that is school starts in August, okay? Now, AUG is always gonna be the start codon, okay? Now notice that the codon pairs up with and again, this region of the tRNA is gonna be called your anticodon. And so this codon, the mRNA, AUG, codes for the aminoacidine MET, or methionine. So that's always going to be the start codon. That just kind of lets it know, okay, this is the starting of my translation process. So basically what happens is once that starts, then the initial, or the remaining parts of the ribosome comes, and now we have your E site, your P site, and your A site. So if you saw the video, it kind of emphasized this whole process, and this was the initiation. So when you're going into initiation, start codon is AUG, okay? Starts with methionine, that's your starting part, all right? And notice we're at a ribosome. Now we can actually go through and read it. And so remember, we're reading the RNA is being moved in this direction, right? Because the 5' end is fed in first. So now we're gonna read this next codon. And so this just shows you an animation, nothing specific about the codons, but basically what you need to understand is that the A site brings over the new amino acid, right? Because these things in purple are gonna be your amino acid. So let's just make sure you're seeing everything that's happening here. Now, this right here is gonna be where you form a peptide bond. So a peptide bond is formed. And the whole thing kind of shifts over, and then the ribosome moves over. So the actual ribosome shifts over. And now you have your E site, which really stands for the exit, or the empty tRNA is going to leave, okay? And then your A site opens up again, and you're going to keep on reading that. So that would be your elongation. So let's talk about what each one of these represents: the coding region, the peptide bond formation, and translocation. So let me show you this quick animation. And this does a good job kind of emphasizing happening here. So right. All right. So if we let this continue, let's see what's happening here. When you let this thing play out, what we're going to see is that, okay, we made your mRNA, and now we're going to see what's going on here at the ribosome. So this is your translation process. So what you're going to see here, this really nice overview, is that as the ribosome is essentially reading your mRNA, this thing in green is your growing polypeptide, okay? Now, what happens here is at a much closer level, or a much closer view, okay? Your ribosome, your base subunit is going to find that AUG, okay? So it's going to read your mRNA sequence. This is the 5' end. It's going to read that mRNA sequence up until it has AUG. AUG is going to code for the amino acid methionine, okay? That lets it give the go-ahead that, okay, now we're going to start protein synthesis, okay? Now, once that happens, the larger subunit is going to come. So the codon, this is your codon, it's going to bond with the anticodon. This is your tRNA carrying the amino acid methionine, okay? Now, skipping ahead, we're going to now read the next codon. Notice that it's being read in triplets, so AUG, the ribosome is going to shift over, and we're going to code for a next one. GAA is going to code for GLU, glutamine. So you can see that that's your P site, this is going to be your A site, and then your E site. So as we let this play out, GAA codes for the amino acid glutamine. This is the tRNA that brings it over. And it's going to show the peptide bond being formed here. Methionine is just going to form a bond with glutamine. And then what's going to happen is this is now an empty tRNA. The ribosome is going to translocate, right, or shift over. This empty tRNA is going to leave. And now what's going to happen is we're going to go through and read these codons. Every 3 nucleotides is going to code for 1 amino acid. So what we're going to follow through with now is UUC is going to code for an amino acid. And you can kind of see UUC codes for lysine, form a peptide bond, it translocates. empty tRNA. This is now going to be the new site, right? And AAU is going to code for another amino acid. So this right here is your primary structure of your protein, right? This is the actual order of your amino acid sequencing. Now watch what happens in this last one, okay? This is going to be what's called your termination phase, and UAA does not code for an amino acid. So let's take a quick look here. UAA is going to represent a stop codon. And so what is going to happen here is it's going to actually bring over a release factor, not necessarily an amino acid, but basically when this release factor hits this stop codon, it's going to signal the termination of this entire process. So now this is going to become your polypeptide, the ribosome disassembles, and your mRNA sequence is cleared up. So that was essentially the termination phase. So just to kind of recap, you're reading your mRNA sequence all the way up until you hit one of those stop codons. So when it asks you here, what determines when a protein is complete, it has to hit one of those stop codons. So stop codons are going to be one of three. And you'll have to memorize these. They're going to be given to you on a chart. UAA is going to be a stop codon. UAG is a stop codon. And UGA. Ways to remember this is UAA is you are away. UAG is you are gone. And then UGA is you go away. Just simple ways to remember. They all kind of associate with stopping. So basically what you need to really emphasize here is if I had an mRNA sequence, could you code for the amino acid? So like, let's say if I had AUG, CCC, GCU, C, UAG, and then let's add some admins at the end, okay? So if this was an mRNA sequence and it said actually transcribe it, let's kind of take a look here. So this is showing you the codon chart, right? So CCC, you might be tempted to go through and read this table and be like, okay, CCC, if this is my mRNA, what's my amino acid sequence? You might be tempted to say proline. However, if it give you a sequence like this on the AP test, don't start there because you cannot start doing transcription until you, you can't start doing translation until you hit that AUG. So how to read this, it's the first base, so you're gonna find AUG. So in terms of this, I don't actually love this chart, so I'm gonna skip ahead to another one because this one has thymines. I don't love that at all. So let me just jump ahead, and I'm just gonna remind you guys that when you get one of these charts, I should have it here. Yeah, sorry. Let me just show you this real quick. So here's one of those charts. If this was, let's say, yeah, here's a better representation. So let's say if you had that sequence CCC, AUG, GGC, UAC, and then UAG, and then CCU, AAA, okay? So on and so forth, right? Like I said, if you found CCC, it would code for proline, but you can't start translation there. You got to start at AUG because that's a start codon. So if this would ask you like, what's the amino acid sequence? It would be methionine would be the first one. The next one would be GGC. So I would have to find this GGC. This is going to code for glycine. UAC, I would find that up here. So I go UAC. That's going to be coding for TYR or tyrosine. And then notice here UAG would essentially be a stop codon. Right? So basically that means that this whole thing is not going to be read. So, and that's okay. So even though I had a relatively lengthy mRNA sequence, I was only 3 amino acids in length. Okay. So remember, this is a codon chart. You need to know how to read them. This is the mRNA sequence and it's telling you what amino acids they're coded for. Okay. And that is translation.